Good morning, everybody, and welcome to the Friday Informatics Institute PowerTalk series. And I'm very happy to introduce today's speaker, Dr. Zechen Chong, Assistant Professor of Genetics at in the UAB School of Medicine, and also an associate scientist in our very own Informatics Institute. He also has appointments in the Minority Health and Research Center and the Center for Clinical and Translational Sciences, CCTS. He completed his Ph.D. at University of Texas, MD Anderson Cancer Center in bioinformatics and computational biology. And he also completed a postdoctoral fellowship. His research interests are in developing algorithms for sequencing data analysis, especially next generation sequencing, addressing current limitations to try to improve accuracy and throughput, and also functions and mechanisms of genetic arrangements and genomics, the relationship between genomics and disease, which is the topic of today's talk, the genomic landscape of cancer in individuals with different BMI.
Okay, thank you, Amy, for the introduction. So my PhD was done in China in Chinese Academy of Sciences. I did my postdoc in MD Anderson. So just a minor correction. So thanks very much for the introduction, and thanks for the opportunity to present our latest work. And I think the virtual format is kind of weird. So I hope you can feel free to ask if you have any questions during this course. I need some communications. Otherwise, I feel a little bit lonely. So yeah, first I would like to introduce a little bit of my lab. Our lab has a mission of making science of biology using bioinformatics. So we have 3 lines of research focus in the lab. So we develop innovative tools, bioinformatics tools, to solve biomedical problems. And we also perform hypothesis-driven data analysis. Starting from a scientific question, we will propose a hypothesis, and based on the consortium data, and we will build computational models to interpret the data. So this is today's topic. And we have extensive collaborations on campus and nationwide. So first, I would like to advertise our lab a little bit. So we developed several tools for long-range sequencing data analysis. So first, we developed a DBreak. This is a tool that was used for long-range stratification detection. It can detect stratifications in both healthy and disease samples, and it can be applied in human and other species. So this work was published early online in Nature Communications this year. And we also have the tool called the Fusion Seeker to detect the gene fusions in cancer genomes. So fusion genes are prevalent in almost all types of cancers. So we developed this tool for cancer fusion detection, and this work was published in Cancer Research. And for the species without a reference sequence, so we develop an assembler. It's called B-assembler. So we can use B-assembler to assemble the circular bacterial genomes. And because of the genome assembly, the long-range genome assembly has some limitations. So there's the lack of effective tools to evaluate the assembly results. So we developed this tool called Inspector to evaluate long-range de novo assembly results. So this work was published in Genome Biology. And we are also involved in extensive collaborations on campus and nationwide. So here's the example. So collaborating with Dr. Louis Shi's lab, we discovered the important role of interferon gamma signaling in shaping tumor infiltration T cell. So the loss of interferon gamma signaling gene is known to related to immunotherapy resistance. So this study helps us better understanding this mechanism and may help improve cancer immunotherapy in the future. Another collaborative effort with Dr. Eddie Young and the GMA Liververse, and we discovered the epigenetic reprogramming of tumor immune microenvironment that is mediated by the gene RNF2. We are continuing working on this, expanding this effort in order to further investigate the underlying mechanisms with the advanced single-cell technology right now. So today, I would like to share a hypothesis-driven work conducted in a group So my student Fengyuan Huang is leading this project. So the project focuses on the obese and the cancer. So we all know that obesity has become a global issue, right? A global health issue. Currently, there are around 40% of adults in the worldwide are considered as overweight or obese. And probably 10 to 15% of them are obese. So obesity and the complications and cost millions and billions of dollars every year. So probably you may heard of it recently. There's a very almost every newspaper was talking about the new weight loss medicine, right? It's very effective. It works very well, but it's cost 13,000 per year per prescription or by I don't know the direct cost. So it's extremely expensive. So World Health Organization uses a metric. It's called body mass index, BMI. So BMI is the way that we can classify people into normal weight, overweight, and obese groups. So in the US, more than 42% of adults are overweight or obese. So we know that in the state of Alabama, the state of Alabama is one of the highest obese states. So especially in minority groups. So this is why UAB has established the Obese Health Disparity Research Center. So this is an urgent topic that we need to investigate. So epidemiology studies, so based on some statistics, so they have identified 13 cancer types that are associated with overweight or obesity. So in both women and men, so esophageal cancer has the highest prevalence. So almost 30% of cancer patients are obese or overweight. While in women, the endometrial cancer consists of 31% cases overweight or obesity. For other cancers, they have a similar trend. For example, kidney, liver, gallbladder, stomach, multiple myeloma, pancreatic cancer, etc. For the cancer locations, the associated cancer type tend to happen in the digestive system and the reproductive system. So the digestive system has something to do with the food, right? We take the nutrition intake, right? So it's reasonable. And the reproductive system have associated with hormones, right? So hormone is also associated with obesity. It's a major contributing factor, as we will show in the next few slides. So how can obesity impact cancer development and progression? So there are several proposed mechanisms. So the obese was caused by the excessive of micronutrients intake. And in the obese, the fat tissue, all the adipocyte cells accumulate and they activate the pathway of JNK, IKK, PKR. These are all protein kinases. So these proteins or enzymes are involved in immune response and inflammation, and also the activation of a Toll-like receptor. A Toll-like receptor are found on the surface of the cells of the immune system, and they are involved in the recognition of various pathogens. So they play a very important role in innate immune response, which is the first line of defense against the invading pathogens. And because of the antiposes, so the antiposes can involve the hypertrophy status, and this can introduce the hypoxia status, and that's further involved in the macrophage infiltration. So the macrophage can release some of the factors like TNF-alpha and interleukin-6. They are all cytokines. So they are produced by macrophage and play a very important role in inflammatory response. And also this is adiponectin. This is a hormone that is secreted by the fat tissue. And low levels of adipose has associated with the insulin resistance. So this promote a state is called a pro-inflammatory status. And this status, and again, together with obesity, can cause endothelial or microvascular dysfunction. All of these together can cause the alteration of in lipid and glucose metabolism, and the inflammatory status, and the insulin resistance, hypertension, etc., and can cause severe metabolic syndromes. Secondly, the obese can alter different types of hormone levels. So there are 4 hormone levels: leptin and insulin, sex hormones, and growth hormones have been proposed with associated with obesity. So leptin signaling is binding to the numerous signaling transduction pathways. So the pathways of interest of importance are like PI3K, the JAK2, JAK-STAT signaling pathway, and the insulin involved in the pathway like PI3K, AKT, mTOR signaling pathway. So this branch can lead to metabolic effects. While on the other direction, the ROS, RAS, RAF, this signaling pathway can cause the metagenic effect. So this metagenic effect is a status that stimulate cell division and proliferation. So it can lead to an increase in number of cells. So for the third point, the obese can affect the tumor microenvironment. Tumor microenvironment is so under the obese status, so many besides the tumor cells, the fat cells accumulated, and it's recruit many immune cell infiltrations, including macrophage, T lymphocytes, nature killer cells, and many other immune cells. So macrophage within the obese tumor, it forms like a crawl-like structure, and also obese can induce the increase of interstitial fibrosis. So all of these create a tumor, obese tumor microenvironment that's different from a tumor microenvironment. So because of this and because of the mechanisms of all of these mechanisms were based on some mouse models or individual genes, and also the previous epidemiology studies, the association between obese and cancer were mainly concluded from epidemiology studies. And overall, the sample size are relatively small in the past. For example, the mouse model and the individual genes analysis. So they have many limitations. So we wonder if there's any molecular differences between obese and the non-obese tumors. So that's our question. So we hypothesize that because obesity can create a unique microenvironment, and this microenvironment can contribute to the molecular differences between tumors, normal weight, obese, overweight, and obese. So here we use the approach, we reanalyze the TCGA samples. So the TCGA samples has some of the samples has a BMI information available. So we start from the somatic mutation, gene expression, DNA methylation, and the immune infiltration from these perspectives. And we compare between the overweight and the obese versus normal weight patients to try to identify the molecular differences between the obese and non-obese patients. So TCGA is a landmark cancer genomic program. So it characterized over 20,000 primary cancer and matched normal samples, spanning 33 cancer types. So many of the main cancer types have been characterized in TCGA database. And it has generated over 2.5 metabytes of genomic, epigenomics, transcriptome and proteomic data. So this is a wealth of resources, and is extremely helpful to many researchers, and especially for the lab like us. We cannot generate our own data. We use public data. So this is a great resources. So here is a summary of the patient samples that has BMI information in the TCGA database. So there are 14 cancer types. So here is the abbreviation according to the cancer types. And overall, there's 2,600 patients in total, and the male and the female distribute kind of evenly. But out of the 13 cancer types, there's only 6 cancer types have this BMI information. But fortunately, these 6 cancer types are the top associated cancer types. For example, endometrial cancer, right? Esophageal cancer, and kidney, liver, rectal cancer, colon cancer. So these are all top cancer types. Although the breast cancer, pancreatic cancer, so they have missed the BMI information, but we can still interrogate some other cancer types like bladder cancer, cervical cancer, etc. So they may have something to do with BMI. We don't know. So we incorporate all these samples in our study. So here, we would like to study the molecular difference between different weight groups, right? The BMI should be the only factor we should consider. However, there are confounding factors that have potential issues. So here we compared between normal weight and overweight, and also normal weight versus obese tumors. For individual catalogic category variables like the histotype, stage, race, smoking status, purity, and age, gender, etc. They're all confounding factors. So we find that between normal weight and overweight or obese, so many of the factors that actually affect they are causing potential issues. For example, the one that marked as red is significant. So to alleviate these confounding factors, so we propose a method. We use the propensity score balancing approach. So this approach was first introduced by my colleague, my friend colleague in MD Anderson. They studied the male-female difference in TCGA samples. So we adopted a similar approach. So the philosophy behind this propensity score balancing approach is that, for example, given a variable, if there's a distribution before filtering, before matching, they have a different shape, right? So we try to do the matching and the balancing, rebalancing, and to rescale it, and so that we can compare them as similar groups. And this is the age, for example, they have different distributions. After matching or rebalancing, so we can compare the age groups individually. So this is the philosophy behind this approach. So this is statistical approach. And see, in our TCGA samples, right, the weight groups are, and because of confounding factors, it's kind of unbalanced. So there's a normal weight and overweight and obese. So we use this approach to match and rebalance and generate a balanced results. And then we compare the normal weight versus obese and overweight and obese respectively. So here the overweight, we were thinking it's kind of like intermediate status. It's a transition status between normal to obese, right? So this is also valuable for us to compare. So we compared different aspects, like as mentioned, point mutations, copy number variations, gene expressions, methylation, and immune features. So here is a BMI distribution among different cancer types. So normal weight, overweight, and obese are labeled in this is brown and light blue and pink respectively. So this color scheme is consistent in the following slides. So overall, you can see it's quite prevalent in the TCGA samples for the samples for the cancer types that has BMI information. And in UCEC, this is endometrial cancer. So if you recall, this is the first highly prevalent cancer types in female cancer patients. So this is consistent with the epidemiology studies. Similarly, several other cancer types has a similar trend. Although sometimes, for example, esophageal is probably the sample the sample size and some other issues. It's not consistent, but it's still overall shows the prevalence in general, the enrichment of obesity and overweight. So the first tier of study is the association BMI with these different cancer types to investigate the somatic mutations, because somatic mutation, the cancer was caused, right, was driven by the somatic mutation. So this is the first tier of resources we should resort to. So we checked the individual cancer types. We found that in the rectal cancer, so BMI is significantly and positively correlated with the tumor mutation burden. And also the a type of a melanoma is called UVM is also is positively correlated with the BMI information. And in the endometrial cancer, although it's not significant, it shows a negative correlation. So the higher BMI, the lower tumor mutation burden, although it's not significant. And when we stratify the mutations into different types, so the somatic mutation contains single nucleotide mutations and indels, right? So we stratify them into indels and single nucleotide variations. So we find for the rectal cancer, it's in the it is in the SNPs, not in the indel categories. The SNPs are highly correlated, but not indels. So we know rectal cancer, the indels are prevalent in rectal cancer because microsatellite instability. And in the esophageal cancer, it's not SNV, but indels are highly correlated and positively correlated with the BMI. So different mutations in different cancers form a mutational signature. So this work was conducted by a Knight, a UK Knight, Dr. Stratton and colleagues at Sanger Institute. So they have summarized around 30 signatures in the COSMIC database. So the philosophy, the rationale behind this is somatic mutations are presented in all cells of the human body and occur throughout the life, right? So there are consequences of multiple mutational processes. For example, the DNA replication, the mutagen exposure, and enzyme modification, right? And the DNA damage repair, etc. So all of these mutational processes form or cause these mutational types. So this is called mutational signatures. So we wonder if obese or overweight environment can cause a difference, can cause or can form different mutational signatures as well. So we checked the signature in different cancer types, and also we check if there are similarities between cancers. So we compared the normal weight with no overweight in individual cancer types, and also normal weight in obese groups in individual cancer types. So we find that in cancer like endometrial cancer, rectal cancer, liver cancer, several of the cancers, these cancers are known epidemiologically, they are associated with obesity. In addition, we also found this is called DLBC, it's a diffuse large B-cell lymphoma, and the CHOL, cholangiocarcinoma. So it's close to the gallbladder, but they have a relatively small sample size. We will not pay too much attention to this one. So in READ, when we compare between normal weight and overweight, we find that in both normal weight and overweight, there are signature 6 and 20 respectively. They are caused by defective MMR. So when we recall the previous slides, we talk about the READ, right? This is the SNPs, SNV, single nucleotide variants, but not indels, are enriched in are highly associated with rectal cancer. So that means the indels probably is not a major contributor to the genomic landscapes or mutational signatures to rectal cancer. But we find that in the obese group, signature 13, and in the overweight group, signature 24, they are highly associated or is overweight or obese bears in the rectal cancer. So these two mutations were formed by extensive C2A mutations, and this is caused by aflatoxin exposure effects. And also the signature 13, this is the C2G mutations. This was enriched in the APOL-BAC pathways. So this is very interesting. And also esophageal cancer. Here we find that the esophageal cancer has a because we identify indels is a highly correlated with the BMI, and here consistently we find that signature 6, this was caused by the defective MMR, is also enriched in the in the obese groups, but not in the normal weight groups. And in addition, we find that another cancer type, DLBC, this is a diffuse large B-cell lymphoma. It's not in the normal weight with the overweight, but in the obese. So there's several signatures popping up in the obese groups. So this cancer previously has not been reported, or is not reported as a highly associated cancer type in the epidemiology study. But we find that there's some studies still showing that there's the statistically significant association between obesity and diffuse large B-cell lymphoma. So this is an example. So another very interesting pattern is in the endometrial cancer type. So you can see the one that marked as gray, this is brown, it's a normal weight bears. So in both overweight and obese weight group compared to the normal group, the normal weight, it shows that the normal weight bears the pattern in the endometrial cancer. So that being said, the mutational signature in the normal weight are bears in the endometrial cancer type. So it's not in the obese. The obese genomic landscape or the microenvironment didn't cause a significant bears in the obese, but in the normal weight. So this is very, very interesting. So this is there is a theory, it's called obesity paradox. So it refers the observation that in certain populations, so individuals with a higher BMI may have a better prognosis and the lower mortality rates compared to the normal or lower BMI categories. I think this is matches to that hypothesis. However, the mechanism underlying this is not clear yet. So next, we check the allele frequency. So high allele frequency, high frequent mutations are potentially driver mutations. So they are very important in cancer genomic study. So we wonder if there are genes that are bears in overweight or obese cancers. So we select the genes that has a highly mutated with a mutation frequency that's larger than 5%. And we check if there's a normal weight bears or obese or overweight bears, the genes can be identified. So we find that this is the genes that are showing either normal weight or obese bears. So esophageal cancer, endometrial cancer, rectal cancer, and the diffuse and the bladder cancer show several different levels of highly bears mutation genes. Especially the esophageal cancer, which has the highest number of bears high frequency genes. So esophageal cancer, if you recall from the epidemiology study, so this is in both women and men, this is the most prevalent cancer types as associated with obesity. So this is called oncoplot. So oncoplot shows the top high frequent mutated genes in esophageal cancer types. And we use a related approach, recapitulated frequency. You can see a majority of these top obese bears the gene. This bears towards obesity. And so several genes of interest is, for example, the FAT4. FAT4 gene is a gene that controls the Hippo pathway. This pathway is associated with obesity. And also there's a top SYNE1 and MDGA2. These two genes have been found to be abnormally methylated in the gastric cancer types, which may affect the gene expressions. So this may be very important genes that we discover from this list. And in the endometrial cancer types, this is it shows a normal weight burst in the high frequent mutated genes. So when we stratify this based on the normal weight and overweight obese burst, we can see the top highly mutated genes tends to burst towards normal weight. So this is consistent with our observation from the mutational signature and some other the mutation frequencies. So it still shows normal weight burst. So it's burst towards normal weight and consistent with the obesity paradox to some extent. So we next use the mute-seq to CV. So this is a tool that to analyze the somatic mutations that discovered in DNA sequencing. So it tries to identify the genes that mutated more frequent than expectation by chance. So it considers several categories. First is it considers the genes has a relatively higher mutation compared to the background mutation. And also it considers the hot spots. So for example, if there is a gene that are enriched, there's many patients or many samples has a enrichment in some hot spots. So this will score the higher. And also it considers the evolutionarily conservation. So if the genes that are conserved across species, right, these genes should show a high because it's under strong purifying selection, so they may functionally significant. So based on this approach, we applied the mute-seq to CV to all of these cancers has BMI information. We apply to normal weight, overweight, or obese, and then we compare the two. So for the top genes, so the green ones are the ones that shared in both normal weight and overweight based on this approach. And the red one, the pink one, are the one that has the overweight or obese. So it's bears towards overweight and obese. And the brown one is the normal weight bears the genes. So we identified from 1 in melanoma and to 137 in endometrial cancers. So different tumors have different number of significant mutated genes popping up. And this is the endometrial cancer and the liver cancer showing here the pattern. So in the middle part that shared between both normal weight and overweight, the genes, we can see some popular cancer-related genes like P10, TP53, right? These cancer genes are popping up and shared between, so they probably have nothing to do with the BMI. However, we find some very interesting genes. So here the UCEC is screwed in the overweight and obese part. One particular gene, this ARID5B. So this is take this as an example. So this genes has something to do with FTO. FTO is not the only, but probably the most important genes and shows that has something to do with obesity. And so these genes can disrupt and lead the increase of IRX3 and IRX5 expression. So these two genes can turn the fat cells into white fat and store the lipids instead of brown fat in the thermogenesis process. And several other genes also show some. I will not discuss in more detail, like the ARAM12, etc. So we notice this is a skewed bias in the obese and overweight in the endometrial cancer. So wonder what genes are they and which pathways are they involved. So we find that this is a pathway enrichment of these significant mutated genes in the endometrial cancer. So we find that the pathways that previously reported, like insulin pathways, the hormone estrogen signaling pathways, growth factor, growth hormone pathways, JAK-STAT signaling pathways, mTOR signaling pathways, and some other the thyroid hormone signaling pathway. All of these are popping up along with several other. So this makes lots of sense based on the previous mechanism study. And the copy number variation is another source of mutation. We also check the copy number variation, its association with individual cancer types. So we find that the thymomer cancer has a highly significant association with copy number variations in both copy number, in total copy number, and copy number loss. And also this one has a CHOL, which has a fewer samples. It shows the copy number gain is significantly associated with the BMI, but the sample size is too small to conclude this. This is significant. And when we check the endometrial cancer type, we find that in the endometrial cancer type, the deletion and amplification. So there's some peaks. So the peaks has a relatively significant enrichment in individual chromosomes. And we check the genes in these chromosomes and see if they are enriched in several molecular functions or pathways. So again, we find that the one that in the normal weight as represented as deletion and obesity represented amplification, they tend to enrich in the pathways like hormone receptor binding pathways, growth hormone receptor binding, and fibroblast growth factor binding, etc. These molecular function categories. So this is consistent with our mutational significant mutated genes analysis. So next, because of genetics may introduce some epigenetics change as well. The epigenetic change can be reflected as the gene expression change. So we check the differential expression genes between the overweight and the obesity compared to normal weight groups. So this is the upper ones is the normal weight, overweight compared to normal weight in individual cancer groups. And the bottom one is the obesity compared to normal weight groups. So esophageal cancer and endometrial cancer, it shows the highest number of differential expressed genes and several other cancer types. But the one that highly associated with obesity shows the most striking results, like liver cancer, rectal cancer, etc. So this is consistent with the epidemiology observation as well. So here is a heat map of top differentially expressed genes in esophageal cancer types. So the left part we sorted based on the weight category. So the left is the brown is normal weight, and here is overweight and obesity. So you can see these top genes. It shows a lower expressed in the normal weight, while it's highly expressed in the obese and highly expressed in obese and overweight groups. And these genes in the bottom part is highly expressed in the normal weight, while it's the lowerly expressed in the obese and overweight in general. So these genes are potentially markers that showing differences between the normal weight versus obese and overweight groups in esophageal cancer types. So because there's too many genes, we wonder if there's some pathways that can be selected that are enriched between the normal weight and the overweight groups. So we did the DEG enrichment analysis. So here is the enriched pathways between overweight and the normal weight groups, and in these cancer types. So the one that marked as the blue are the pathways that are enriched in the DEGs that tend to be enriched in the overweight groups, while the ones that in brown is the genes that DEGs tend to be enriched in the normal weight groups. So we can see in the overweight group compared to the normal weight group, majority of the differential expressed pathways are the genes that are highly expressed in the overweight group. And similarly, we compare the DEGs between the obese and the normal weight groups. The pink ones are the genes are differentially expressed or enriched in the pathways that are biased towards obesity, and the brown is biased towards normal weight. So similarly, we show, you can see the majority of the cells are pink, right? That means the genes that are differentially or enriched in the obese pathways, the obese is highly expressed in these pathways. And these pathways include the one that we mentioned, like mTOR signaling pathway, the insulin resistant pathway, insulin signaling pathway. So the classical obese related pathways popping up. In addition, we found several other interesting pathways, like the cell cycle, mismatch repair, DNA replication, chemical signaling. So many other pathways also popping up. These pathways may shed light on the underlying mechanism between obese and normal weight groups as well. So we checked if the DEGs that has a clinical effect. So we checked the percentage of DEGs if the associated with the survival time in each cancer types. So here are the results. In individual cancer types, we find the genes, we separate them into overweight and group, overweight and obesity group, and also the upper regulated and down regulated. So this is a different cancer types show different fraction. And here is the esophageal cancer. Although the esophageal has a high prevalence, but it's not too many genes that the esophageal has the highest number of DEGs, right? But only like 5 to 8% of the genes show associated with the survival. So some of the top genes that regulate upregulated in obese groups, and they show survive. They associated with the poor survival in esophageal include the following ones, like the FOS and the BRCC3 and the IL-13RS1. So this FOS gene is a gene that regulates inflammation and metabolic and stress response. And BRCC3 is a gene that involves DNA repair and NF-kappa B signaling. And they also, their alterated expression can lead to inflammation and tissue damage. And this gene IL-13RA1 involves the inflammation and the immune response as well. So they are all related to the obese contribution to cancer to some extent. And the most strikingly, we found here the endometrial cancer type. So endometrial cancer type, as we showed, and this shows the pattern of obese paradox to some extent. So this cancer type has the highest number of fraction of DEG, the renal expressed genes. Like here it's almost 30%, 20 to 30% of the DEGs associated with the survival time in endometrial cancer type. So when we stratify the endometrial cancer type and see if it's the regulation, if the expression has a better survival or worse survival, when we stratify them genes in the endometrial cancer type, the total number shows in the first line. So there's downregulated genes and upregulated in obesity, and downregulated in overweight, and upregulated in overweight. And the number of genes, here the percentage, 20 to 30% is listed here. And we find that very interestingly, the one that has higher expression, so the one that are upregulated in obesity, actually has a better survival. And this is true for the overweight as well. So 86% and 78% respectively. So these genes are highly expressed in overweight and obese and have a better survival. And also the ones that are downregulated in obese or downregulated in overweight. So a large fraction of these genes shows a better survival in the lower expression. So that means the DEG in obese and overweight overall has a high fraction of genes that showing better survival. So that's evidence of the obesity paradox, right? A better, so the obese environment actually contribute a better survival and less mobility. So here's the overall results. So the upregulated genes is marked here. So this is obese upregulated genes, and this is overweight highly expressed genes. So in both these two groups, the overall survival is better in the obesity and overweight group in both comparisons. So that reflects this pattern. So we wonder why was that case. So when we go back to the differential expressed genes, the enrichment pathways. So as you saw, majority of these pathways, right, they are highly expressed in the obese groups for many other cancer types. But the endometrial cancer type shows the opposite pattern to some extent. You can see the majority of the pathways, all the genes are enriched in this normal weight bears. So that means that it's highly expressed in the normal weight. And when we check the pathways that enrich in these normal weight bears, the DEGs, they show that the cell cycle mismatch repair, DNA replication, and the P53 signaling pathway, mTOR signaling pathway, etc. These pathways are popping up. So that means, the normal weight has a higher mutation burden, and the mismatch repair actually has highly expressed enrichment in the mismatch repair. So they tend to fix the density, so they try to keep the cell alive, right? So these cells are growing better in the normal weight. So that's the reason we were thinking that caused this obesity paradox. And actually these promote tumor proliferation in the normal weight to some extent. So methylation is another pattern. Methylation is always consistent with gene expression. So we also compare the methylation pattern between the normal weight versus overweight and normal weight versus obese group. So a hyper mutated methylated pattern, because methylation tends to repress the gene expression. So here we can see overall there's more normal weight bears, right? That means it's hyper methylated in the normal weight group. It's consistent with our gene expression pattern. So we have observed that a higher number of obese bears the pattern, right? This is consistent. But there's a liver cancer. It shows so it's in the liver cancer in the obese groups. It also shows hyper mutated in the obese group, but not in the normal weight group. That means that it's probably not the methylation that regulates the gene expression in liver cancer obese groups, but there's some other mechanism we don't know yet. So the next very important topic is the tumor microenvironment. So the tumor microenvironment from the first few slides I have introduced. So the tumor cell can, under the obese microenvironment, it introduce the immune cell infiltration. So different immune cells tend to be enriched in this. So because the TCGA has no such information, so we use a state-of-the-art, it's called tumor infiltration deconvolution analysis. So we use some single cell data, and we use to deconvolutionize the cell fraction, so we can characterize the immune cell fraction in the tumor samples, in the TCGA samples. So we find that in the liver cancer and the cervical cancer and the rectal cancer, so there's several immune cell like leukocytes, lymphocytes, etc. These shows a higher infiltration in the obese group. So this is consistent with the higher infiltration size cells infiltration in the obese environment, microenvironment. However, in the endometrial cancer, we will observe the like the helper T cells and the CD4 T cells. So it's a down, it's a has a lower enrichment in the obese groups, and there's a higher enrichment in the in terms of Treg cells enrichment in obesity groups. So we know Treg is a suppressed immune response. So this is in the endometrial cancer, it shows something different. This is probably true compared to the overall when we analyze the endometrial cancer versus the other cancer types. The obesity paradox may also hold here. But on the other hand, because of the tumor microenvironment can be caused by different resources, right? Tumor itself can recruit the immune cells to some extent. And also the obese microenvironment can recruit different cell types of inflammation and can activate the immune cell infiltrations landscapes difference. So something like that. So it's quite complex to conclude if how the immune cells can contribute to the obese cancer environment, microenvironment. We also checked the immune features. So the immune features include the TCR and the BCR enrichment and the SNV new antigens, wound healing, etc. All of these features. So similarly, we find that different cancers show different patterns. So the one that marked with solid are the significant ones and has a false discovery rate that's smaller than 0.2. So the endometrial cancer, we find there are several features biased toward the normal weight group. So this is consistent with our previous endometrial. So it's always normal weight biased pattern. While for some other cancer types, like the esophageal cancer type and liver cancer types, so there's both bias for different immune features in normal weight and obese. So that's implies the immune microenvironment is very complex and it needs a better study. It needs further study to better explain this pattern. So to summarize, we proposed a study to study the different molecular differences between obese and normal weight group and overweight versus normal weight group. And we used the propensity score balancing approach to overcome the confounding factor issues. And we found that somatic mutation burdens and mutational signatures are different between overweight and obese compared to normal weight groups in several cancer types. And the differential expressed genes analysis significantly find some pathways and some of the DGs can be served as potential prognostic markers because they have a significant difference in terms of survival. And here, the endometrial cancer type seems to benefit from obese. So despite this has an extra burden. So the obesity seems to be beneficial for endometrial cancer types. So this is consistent with the obesity paradox. So this can be reflected from endometrial cancer types. And finally, the obesity and cancer makeup tumor microenvironment is very complex. So it may need a further study to further explore this direction. So in the future, we would like to follow up on the experiment validation of these discovered markers. And we will further investigate how obese can contribute to the endometrial carcinoma. And for the tumor microenvironment, I think the deconvolution study is limited because of the mathematical model may not be accurate enough, and also the tumor collection and many other issues. So it's not ideal results. So based on the single cell sequencing, probably that's the best way that we can touch this potential issue, the microenvironment issue. And more samples with genomic data are needed to understand this because we can see from the sample size distribution. There's many cancer types still has a limited sample size. So this is a further direction that the field should be, the community need to pay attention to. So finally, I would like to thank my lab members. Feng Yuan is my student. She's leading this project. And my former postdoc, Peng Xu, also involved in part of this. And my students, Yu Wei and Kai Li, also involved part of the projects. And my collaborator, Min Gao, Xin Yang, and Zong Liang also contributed to some extent to this project. And we use the consortium data, TCGA and SCGC, to perform all of the analysis. So the work was sponsored by Informatics Institute and the OHDRC and GMS. And thank you for your attention. And I would like to take any questions.
Like how it's so translational, covering all these aspects, going all the way from genomics analysis and bioinformatics to clinical medicine. Just kind of wondering, what other directions or conditions might be next that you might be looking at?
Yeah, I think the obese cancer association is a very important topic. And NCI has some mechanisms. There's some FOA or FRA. Yeah, they have special interest in this topic. But I think the molecular and genomic approaches to explore this direction is limited so far. And we have the diabetes center, right? We have OHDRC. So I think UAB has a unique opportunity to pursue this direction. That's why I launched this project. And I think like we have a group of people. Alabama is a highly obese state, right? And there's a minority issue, and so many of these could be great opportunities to pursue this obese cancer and some other health disparity issues. So that's oh
yeah, thanks for reinforcing that, highlighting that. You're right, that the economic medical impact, and plus we know even that obesity is a risk factor for COVID as well. Any final burning questions from the audience, since I realize that we're at the end of our time?
Yeah, I just want to, it's not a question. I want to thank Zechen for doing this. And I also want to point out that, this is informatics research first and biomedical research second. You've developed methodologies that can be applied to a lot of different things, and then apply them to solve a particular problem or address a particular condition, or, and that's what the Informatics Institute is all about, is having research of that kind. And working in a domain area is what informs us and helps us develop new ideas and say, gee, this would work better if we changed our methodology, and then we can apply to other things. So I just want to thank you very much for staying the course in bioinformatics research.
Yeah, of course. I think that's very good command, and a very nice command. So when we develop tools, and people will ask, what these tools can be applied, and are they useful or not? So actually, this is an example of for this aspect. And so many of the bioinformatics tools that develop is to address the significant or important biomedical problems. But sometimes probably the tools develop far beyond the development of biological problems. So it needs there's a gap, but we are trying to fill the gap to some extent. That's what we are working on. Yeah.
Okay.
Collaborated with you as well, and good to know you. And if anybody else is interested in this kind of work or has any ideas, please, we're very much about collaboration, so please get in touch with Dr. Chong. And once again, thanks for a wonderful talk, and we hope to see you at the next Power Talk. Have a great weekend.
Thank you, guys.
Thanks.
Bye-bye.